evening. So, as Sarah mentioned, I'm going to give more of a historical perspective on the whole question of marine biology and science that we can learn from the deep sea. And so I want to invite you this evening to go on a journey with me, both into the depth of the ocean as well as back in time, to understand how marine organisms were important model systems at the outset of several very different fields of biology. And in this journey we'll be meeting some Nobel Prize winning squids as well as some very frisky sea urchins. So I hope it's going to be an exciting 10-minute talk. But the first animals that we're looking at are these squids. And they seem incredibly alien to us, and they're fascinating in many respects. But most importantly, for the focus of my talk this evening, is that they were really important model organisms in the development of neurobiology. And so this is kind of going back to what Federico talked about with the brain and brain development. And I'm sure you all know that neurons and nerve cells are especially important in essentially everything. And they're important in our functioning of our bodies, and they're important now in your brains, trying to listen to me while at the same time trying to think whether you want to have another beer. And they communicate via electrical signals. And this has been known for a long time. Actually, since the year 1902, when a German scientist by the name of Julius Bernstein for the first time saw electrical impulses stimulating the neural activity. But he wanted to measure the electrical signals of a single cell, but he couldn't, because he was working on frogs. And frogs have these teensy tiny neurons, and so it's impossible to measure. Because if you think back to high school, this is what the machines look like that you used to measure electricity. These are these voltmeters, and they have these two poles that you stick into whatever circuit your teacher told you to measure in high school. But actually, if you want to measure electrical signals in a cell, the system is very similar. And if you don't have a fancy microinjection setup, like what Federico showed us before, and they didn't in 1902, it was absolutely impossible to measure. And so, for almost 30 years, nobody could measure electrical signals in neurons until a young British zoologist by the name of John Zacharias Young was sitting in Plymouth at the harbor in the morning and watching fishermen bring in boatloads of squid. And because he was a zoologist, he did what zoologists do, namely dissect. And so he cut open the squid, and what he saw really surprised him, because these squids have a gigantic neuron which controls the muscle activity of their mantle, which is what makes them swim. And these neurons, they're about this. So if you stimulate these neurons by electricity, even after you cut off the head of the squid and cut open the mantle, this is what happens. So if you put electricity to this neuron, the mantle twitches, so it's still active even after the squid is officially dead. And because this neuron is so big, you can actually dissect it with a normal set of tweezers and scissors. And it's about as big as a boiled spaghetti, and apparently from handling it, it's about to handle like a very overcooked spaghetti as well. And it's 1,000 times thicker than a human neuron. So this is much more handleable for a scientist at this time. And as well as being huge, it's also super robust. So you can isolate this neuron from the squid and it will remain active. So you can stick needles into it now, once you've gotten it out of the sun and it's lying in the dish like this here. You can stick needles into it, you can measure the electrical potential across the cell membrane. You can take out the contents of the neuron, analyze them, or you can put other stuff back in and see what effects this has. And so this, in the 1930s, this discovery of these giant squid neurons started the heyday of neurobiology. But this was even improved when in the 1950s, people in Chile discovered that their fishermen were bringing aboard these huge Humboldt squid. They are one of the largest squid species. They grow normally up to 2 meters in length, and they have even bigger axons. So Chile became the mecca for neuroscientists, and among them were two people Andrew Huxley and Alan Hodgkins, who actually in 1963, so 10 years after they started their work on neurobiology in squid, got a Nobel Prize for their results. So this was very quickly recognized how big the impact was. And Hodgkins always used to joke afterwards in all his interviews that the squid were actually the people who should have gotten the prize. But of course, as it is with scientists, in the moment nobody thinks about the model system, so the squids were overlooked. And with this thought, we'll end the view on the squid biology and move on to sea urchins. And these are very weird animals. They are these dome-shaped balls of spikes who live on the seafloor, and they're also incredibly widespread. So you'll find them in tidal pools in the French coast and in the deep sea beneath the Antarctic sea ice. And they have been used in biological research for 150 years, mainly because very early it was discovered that you can make them lay eggs. So you can mistreat them a bit, inject them with some salt water, and then they'll just release tons and tons of eggs. And this is what you can see here. You have an urchin on top of a glass beaker, and all this yellow stuff here at the bottom is millions upon millions of eggs. And these eggs have two very special qualities. They are very big, and at the same time very transparent. So even in 1875, Heideblerg, the scientist named Heideblerg was able to put them under this pretty crap microscope compared to modern standards and see the fertilization. And what he actually saw is something like this. So here you have the large egg cells, and then you see these tiny dots moving around. These are the sperms, and then fertilization happens. And this was in 1875 the first time somebody understood how sexual reproduction worked. Until then, nobody had any idea, and they thought in the sperm there's a homunculus, like a tiny human being that just grows in size. And now it seems very trivial to us, but it was a huge step forward in understanding, and it opened up a completely new field of biology, because now it becomes a question of how do we go from one cell to an organism which has millions upon millions of cells. And this process, of course, called cell division. And this is here in early embryos. You have a fertilized egg, and this will then divide. So one egg gives 2 cells, gives 4 cells, gives 8 and 16, and so on, until you get a full-grown organism. And this is actually, and this was really key, the sea urchins, because they produce so many eggs. They were very helpful in actually understanding this process of cell division and the cell cycle. And in 1983, Tim Hunt actually used sea urchins to understand and to discover the key factor controlling the cell cycle progression. And this story again ends with a Nobel Prize, which in 2001 Tim Hunt got along with Hartwell and Nurse for the discovery of the cell cycle controlling machinery. And nowadays we still use sea urchins in research, and we can make these beautiful pictures of the cell division machinery, which look like fireworks in the cell. You can see in blue the DNA, and in gold the microtubules, which split up the DNA into 2 equal parts. And while you further enjoy looking at these pictures, I want to summarize the points of what made these two very different animals such good model organisms. And I think it's become very clear that the first factor was that they were very large compared to most other animals in this specific respect. So the squid had a large neuron, and the sea urchin had a large egg. And at the same time, while they were large, it was really important because back then we can't really imagine how bad the microscopes were. So it was impossible to see anything, and so the bigger the better. And at the same time, both the eggs of the urchin and the neurons of the squid are incredibly robust. So these eggs, they'll sit happily for weeks in seawater at room temperature, because that's what they do normally. They just sit around until they get fertilized and then develop. So this was also important in this very low-tech times when keeping the model systems you were working on was a real problem. And there's another point that I noticed while I was preparing this talk and looking for pictures that connects both the squids and the urchins. And that is that apparently they make a very nice dinner. So grilled squid and sea urchin soup are apparently delicious, And I'll leave you to puzzle out how big the impact of this factor was when the scientists were deciding to do research on these animals. Thank you.
Thank you, Johanna. I think your talk is going to be complementary to mine, as we both work in the deep. So, does anybody have a question for Johanna? No.
I won't bite.
You know, Squids.